friends, welcome to a new session on molecular biology. Here we discuss types of mutation. Mutations are sudden, stable and discontinuous heritable changes in the genetic makeup other than those produced by Mendelian segregation and recombination. They appear almost abruptly without apparent intermediate stages. Essentially, mutations involve three sets of events, namely 1. Changes in the genetic material, 2. Process which produce such genotypic changes, 3. Phenotypic alterations produced by genotypic changes. Changes in the genetic material may involve either single base changes or large scale rearrangements. The term mutation was first introduced by Hugo de Vries in 1901 to denote the phenotypic variations he observed in evening primrose. Following Hugo de Vries, Morgan and his associates could observe mutational changes in the eye color, wing shape, body color, etc. of Drosophila melanogaster. H. J. Muller, who first demonstrated the mutagenic effect of X-rays and was awarded Nobel Prize in 1949. His work was published in 1927 in a paper entitled Artificial Transmutation of the Gene in Science Journal. A mutant is an organism or a new genetic character arising or resulting from an instance of mutation, which is a base pair sequence change within the DNA of a gene or chromosome of an organism. The artificial induction of mutation is termed as mutagenesis and the mutation inducing agents are called mutagens. Mutation is the ultimate source of genetic variation, which in turn is the fundamental mechanism of speciation. In fact, mutation is not a strong force in changing the gene pool. Still then, it provides sufficient grit for ever-rolling mills of biological evolution. Mutations introduce new alleles and new allelic combinations into the population and thereby enhance the evolutionary potentiality of organisms. Most mutations are not beneficial, since any change in the delicate balance of an organism having a high level of adaptation to its environment tends to be disruptive. As the environment changes, however, mutations can prove advantageous and thus contribute to evolutionary change in the species. Part 1. Classification of mutations. Mutations are variously classified based on different aspects such as their origin, the type of cells in which they occur, the nature of their effect, their direction, intensity of the expression and so on. Some of the major kinds of mutations are the following, such as 1. Somatic and germinal mutations. Mutation may occur in any cell and at any state in the cell cycle. The immediate effect of the mutation and its ability to produce a phenotypic change are determined by its dominance, the type of cell in which it occurs, and when it happens relative to the life cycle of the organism. Somatic mutations arise in the somatic cells and passed on to other cells through the process of mitosis. The effect of these mutations depends on the type of the cell in which they occur and the developmental stage of the organism. If it occurs early in development, larger the clone of the mutated cells. Somatic mutations are not transmitted to the next generation through sexual reproduction, but perish with the death of the organism. Hence, they are non-heritable mutations and they do not get established in the population. Thus, somatic mutations have no evolutionary significance. However, in some plants, they can be passed on to the progeny through vegetative propagation. To maintain this mutation, the individual containing the mutation must be cloned. So, in such cases, they are used for the production of new varieties of plants. Seedless grapes, navel oranges, delicious golden apples, horticultural varieties, flowering plants, etc. are produced by applying somatic mutations. Two examples of somatic clones are navel oranges and red delicious apples. Horticulturists first observed the mutants. They then grafted mutant branches onto the stalks of normal trees. After the graft was established, cuttings from that original graft were grafted onto tree stalks. In this way, the mutation was maintained and proliferated. Descendants of the mutant type are now widespread in apple orchards and orange groves. The formation of white scars in the red eyes of Drosophila male is a somatic mutation. The bud mutation occurring in the meristematic cells of axillary buds is another example. Germinal mutations are the mutations occurring in the cells of the germ line, that is, the germ mother cells, gametes and zygotes. They are heritable and hence transmissible from one generation to the next. Consequently, they get established in the population, serving as raw materials for speciation and evolution. The earliest recorded dominant germinal mutation in domestic animal was that observed by Seth Wright in 1791 on his farm by the Charles River in Dover, Massachusetts. Wright noticed a peculiar male lamb with unusually short legs in his flock of sheep. It occurred to him that it would be an advantage to have a whole flock of these short-legged sheep which could not jump over the low stone fences in his New England neighborhood. Wright used the new short-legged ram for breeding his 15 ewes in the next season. 2 of the 15 lambs produced had short legs. Short-legged sheep were then bred together and a line was developed in which the new trait was expressed in all individuals. The mutation that gave rise to the short-legged sheep was obviously of the germinal type because the cell carrying the mutation had the capacity to reproduce the entire organism. Germinal mutations have since been described in a variety of animals and plants. The short-legged Ancon sheep formed by the recessive mutation of the normal type is a classical example of germinal mutation. The second one is gametic and zygotic mutations. Gametic mutations occur during gamete formation. They may cause the development of mutant traits in all the gametes. Zygotic mutations occur during the first division or the subsequent divisions of zygote. This results in the development of mutant traits only in those cells which are involved in the process. So, zygotic mutations may produce mosaic organisms with normal cells and mutant cells. The third one is chromosomal and gene mutations. Chromosomal mutations are abnormal structural and numerical changes of chromosomes. Mutations of autosomes are called autosomal mutations and those of sex chromosomes are called sex-linked mutations. Sex-linked mutation can be X-linked or Y-linked. Fragile X syndrome is a genetic condition that causes a range of developmental problems including learning disabilities and cognitive impairment. Mutations in the FMR1 gene causes Fragile X syndrome. The FMR1 gene provides instructions for making a protein called Fragile X mental retardation 1 protein or FMRP. All cases of Fragile X syndrome are caused by a mutation in which a DNA segment known as the CGG triplet repeat is expanded within the FMR1 gene. Fragile X syndrome is inherited in an X-linked dominant pattern. Abnormal structural changes of chromosomes are called chromosomal aberrations, and abnormal numerical changes are called ploidy changes. Chromosomal mutation affects the entire chromosome and the whole set of genes contained in it. So, they are often called gross mutations. Autosomal trisomy of 21st chromosome in human leads to Down syndrome. Sex chromosomal trisomy may lead to Klinefelter syndrome, and monosomy in humans causes Turner's syndrome. Gene mutations are abnormal alterations in the chemical makeup and molecular organization of the genes. So, they affect only the individual genes located in small portions of chromosomes are generally called point mutations. At the DNA level, there are two main types of point mutational changes: base substitutions and frame shift mutation. Base substitutions are those mutations in which one base pair is replaced by another and can be divided into two subtypes: transitions and transversions. Base pair additions and deletions are collectively referred to as frame shift mutations. 4 different transitions and 8 different transversions are possible. Frame shift mutation introduces new alleles and new allelic combinations into the population and thereby enhances the evolutionary potentiality of organisms. Minor changes in the coding sequence affect gene regulation, thereby affects both biochemical and developmental functions which play a major role in the evolution of morphological forms. In this figure, you can see the process of transition versus transversion. In transition, a purine base will be converted to another purine base. In transversion, a pyrimidine base will be replaced by another purine base. Although there are twice as many possible transversions because of the molecular mechanisms by which they are generated, transition mutations are generated at higher frequency than transversions. As well, transitions are less likely to result in amino acid substitutions due to wobble and are therefore more likely to persist as silent substitutions in populations as single nucleotide polymorphisms or SNPs. The fourth one is micro and macro mutations. Micro mutations produce small and rather inconspicuous phenotypic effects which may not be easily distinguishable from the effects produced by environmental factors. On the other hand, macro mutations produce strikingly prominent phenotypic changes. In general, changes in the quantitative traits governed by oligogenes are macro mutations. Micro and macro mutations are likely to be ill adapted until compensating changes occur in other gene loci. The fifth one is biochemical mutations. Mutations which curtail the production of enzymes and thereby disrupt normal metabolism. This in turn causes inborn metabolic errors. Inborn human disorders such as phenylketonuria, alkaptonuria, albinism, etc. are caused by biochemical gene mutations. Phenylketonuria is a hereditary condition in which the patient experiences a lack of phenylalanine hydroxylase in his system. Phenylalanine hydroxylase is the enzyme used to break down phenylalanine, a necessary protein in the body. Most people have more than enough phenylalanine present in their systems. What isn't used directly by the body is turned into tyrosine by phenylalanine hydroxylase. Tyrosine is an important chemical which is used to build things such as hormones, which are chemical messengers for the brain, and melanin, which is a skin pigment. Because of their lack of phenylalanine hydroxylase, phenylketonuria patients are found to be lacking in tyrosine and the important hormones and chemical that it builds. Due to the lack of melanin, untreated patients are described as having light pigmentation. Biochemical mutations have a lesion in one specific step of an enzymatic pathway. For bacteria, biochemical mutants need to be grown on a media supplemented with a specific nutrient. A nutritional requirement mutant strain of microorganism that will proliferate only when the medium is supplemented with some specific substance not required by wild type organisms is called as auxotrophic mutants. Researchers have used strains of E. coli auxotrophic for specific amino acids to introduce non-natural amino acid analogues into proteins. For instance, cells auxotrophic for the amino acid phenylalanine can be grown in the media supplemented with an analog such as para-azidophenylalanine. Often, though morphological mutants are the direct result of a mutation in a biochemical pathway. In humans, albinism is the result of a mutation in the pathway which converts the amino acid tyrosine to the skin pigment melanin. Cretinism results when the tyrosine to thyroxine pathway is mutated. Here, the figure shows the phenylalanine metabolic pathway. Phenylalanine is a primary amino acid that is abundant in dietary protein. Its main metabolic pathway yields the amino acid tyrosine, which is involved in the production of melanin pigments. Defects of enzymes responsible for interconversion of metabolites in the pathway are the cause of 3 well-studied single gene inborn errors of metabolism, which are phenylketonuria, albinism or melanin deficiency, and alkaptonuria or excess homogeneic acid. The next type of mutation is lethal mutations. Lethal mutations are harmful mutations which can block the normal development and vital functions of cells or organisms and cause their death before maturity. Sickle cell anemia is a human disease caused by lethal gene mutation. Mutant lethal genes are of 3 major kinds, namely recessive lethal, dominant lethal and conditional lethal. Recessive lethal genes express their lethal effect only in their homozygous condition, for example, in the case of sickle cell anemia. On the other hand, the lethality of dominant lethal genes is manifested both in their homozygous and heterozygous condition. For example, in the case of epiloia gene in human beings, which can cause abnormal skin growth, several mental defects and multiple tumors leading to premature death. Thus, it becomes clear that dominant lethal will not be maintained in the population. Examples for lethal recessive mutation. Sickle cell disease is a disorder that affects the red blood cells which use a protein called hemoglobin to transport oxygen from the lungs to the rest of the body. Normally, red blood cells are round and flexible, so they can travel freely through the narrow blood vessels. The figure A shows the effect of mutations in the beta globin gene. Figure B shows the phenotypic changes which occur as a result of the recessive mutation. The mutation causing sickle cell anemia is a single nucleotide substitution, that is from adenine to thymine in the codon for amino acid. The change converts a glutamic acid codon GAG to a valine codon GTG. The form of hemoglobin in persons with sickle cell anemia is referred to as hemoglobin S. The hemoglobin molecule has 2 parts, an alpha and a beta. Patients with sickle cell disease have a mutation in a gene on chromosome number 11 that codes for the beta subunit of the hemoglobin protein. As a result, hemoglobin molecules do not form properly, causing red blood cells to be rigid and have a sickle shape. These sickle shaped cells get stuck in the blood vessels and are unable to transport oxygen effectively, causing pain and damage to the organs. The next mutation we are going to discuss is conditional lethal mutation. Conditional lethal mutant is lethal under one set circumstances, but not under any other set. Conditional lethal have been detected in Drosophila, Neurospora, barley, maize, etc. Nutritional requirement mutants are good examples. Temperature sensitive mutants are conditional lethal mutants that have made it possible for geneticists to work with genes that control vital functions of the cell, such as DNA synthesis. Many temperature sensitive mutants are completely normal at 25 degrees Celsius, but cannot synthesize DNA at 42 degrees Celsius. Presumably, temperature sensitive mutations results in enzymes with amino acid substitutions that cause protein denaturation to occur at temperatures above normal. Thus, the enzyme has normal function at 25 degrees Celsius, the permissive temperature, but is non-functional at 42 degrees Celsius, the restrictive temperature. The interesting thing about conditional lethal mutants of E. coli that cannot synthesize DNA at this restrictive temperature is that they have a completely normal DNA polymerase I. From the information, we infer that polymerase I is not the enzyme E. coli normally uses for DNA replication. When an organism with the conditional mutation of polymerase I was isolated, it was able to replicate its DNA normally, but unable to repair damage to the DNA. This led to the conclusion that polymerase I is primarily involved in repair rather than replication of DNA. Conditional lethal mutants thus show genetic analysis on genes otherwise impossible to study. The next mutation is dominant and recessive mutations. Based on the relation of the mutant alleles with their wild forms, 4 kinds of mutations can be recognized, namely dominant, recessive, codominant and partially or incompletely dominant mutations. In relation to the wild type alleles, the mutant alleles of dominant mutations are dominant, those of recessive mutations are recessive, those of dominant mutations are neither recessive nor dominant, and those of incompletely dominant mutations are only partially dominant mutations and are only partially dominant. Wild type alleles typically encode a product necessary for a specific biological function. If a mutation occurs in that allele, the function for which it encodes is also lost. The general term for these mutations is loss of function mutations. The degree to which the function is lost can vary. If the function is entirely lost, the mutation is called a null mutation. It is also possible that some function may remain, but not at the level of the wild type alleles. These are called leaky mutations. Loss of function mutations are typically recessive. When a heterozygote consists of the wild type allele and the loss of function allele, the level of expression of the wild type allele is often sufficient to produce the wild type phenotype. Genetically, this would define the loss of function mutation as recessive. Alternatively, the wild type allele may not compensate for the loss of function allele. In those cases, the phenotype of the heterozygote will be equal to that of the loss of function mutant, and the mutant allele will act as a dominant. Although it would be expected that most mutations would lead to a loss of function, it is possible that a new and important function could result from the mutation. In these cases, the mutation creates a new allele that is associated with a new function. Any heterozygote containing the new allele along with the wild original type of allele will express the new allele. Genetically, this will define the mutation as a dominant. This class of mutations are called gain of function mutations. The next mutation we are going to discuss is silent and neutral mutations. Silent mutations are the gene substitution mutations which do not produce any abnormal alteration in the gene product and the phenotype. They usually occur in the amino acid sequence encoded by the gene and is not changed, and such mutation is called as silent. Silent mutations tend to accumulate in the DNA of organisms. Then they are known as polymorphisms. Here in this figure, you can see a silent mutation occurring at the phenylalanine codon. In the next figure, you can see the silent mutation in the lysine codon. Neutral mutations are the mutations which do not bring out any selective advantage or disadvantage to the organism. Neutral mutations that are inheritable and not linked to any genes under selection will either be lost or will replace all other alleles of the gene. Neutral mutations are also the basis for using molecular clocks to identify such evolutionary events such as speciation and adaptive or evolutionary radiations. Mutations that alter the amino acid sequence of the protein but does not change its function as replaced amino acid is chemically similar, or the affected amino acid has little influence on protein function. In this figure, you can see the effect of neutral mutations on leucine, which has been converted to isoleucine. The next mutation which we are going to discuss is back mutation and suppression. The mutation of a wild type gene to a form that results in a mutant phenotype is usually referred to as forward mutation. The wild type and mutant represent two different phenotypes. For example, we consider the two alleles controlling brown and blue eye color in humans both to be the wild type. However, in a population composed almost of brown-eyed individuals, the alleles for blue eyes might be thought of as a mutant allele. In any case, the mutation events are often reversible. That is, a subsequent mutation may occur that restores the original wild type phenotype. This is referred to as back mutation. Back mutation may occur in two different ways: restoration of the original phenotype, which may occur by a true back mutation at the same site in the gene as original mutation, restoring the wild type nucleotide sequence, or by the occurrence of a second mutation at a different location in the genome which somehow compensates for the first mutation. Mutation of the latter is called suppressor mutations. Suppressor mutations may occur at distinct sites in the same gene as the original mutation or in different genes. Suppressor mutations can be distinguished from true back mutations by back crossing the revertants to the original wild type. If the wild type phenotype was restored by a suppressor mutation, the original mutation will be present and can be separated from the suppressor mutation by recombination. Suppressor mutation alleviates or reverts the phenotypic effects of an already existing mutation. Genetic suppression therefore restores the phenotype seen prior to the original background mutation. Suppressor mutations are useful for identifying new genetic sites which affect a biological process of interest. They also provide evidence between functionally interacting molecules and intersecting biological pathways. Suppressor mutation occurs in two different ways: intragenic and intergenic. Intragenic suppression results from the suppressor mutations that occur in the same gene as the original mutation. Intragenic suppressors are mutations within the same gene that restore function of the mutant gene product. Suppressor mutations are useful for identifying new genetic sites which affect a biological process of interest. They also provide evidence between functionally interacting molecules and intersecting biological pathways. Intragenic suppression include same site replacement, compensatory mutation, alteration in splicing and reversion of dominant mutations by cis knockout. Intragenic suppression by same site replacement can occur simply by mutating the sequence back to the wild type. However, intracodonic suppression can also occur if a different base change can result in a functional coding sequence. An example is provided by a glutamine to proline mutant of DPY31 encoding a collagen processing protease which has been repeatedly reverted either by back mutation from proline to glutamine or by the only other acceptable change in this codon, that is proline to serine. Mutations resulted by either a single base insertion or a single base deletion could be suppressed or restored by a second mutation of the opposite sign as long as the two mutations occurred in the same vicinity of the gene. This led to the conclusion that genes needed to be read the specific reading frame and a single base insertion or deletion would shift the reading frame, which is known as frame shift mutation, in such a way that the remaining DNA would code for a different polypeptide than the one intended. The second mutation of opposite sign suppresses the original mutation by restoring the reading frame as long as the portion between the two mutations is not critical for protein function. Francis Crick used suppressor mutations to determine codon size. 1 and 2 base insertions or deletions of the same sign resulted in a mutant phenotype. Deleting or inserting 3 bases could give a wild type phenotype. In this figure, you can see the restoration of the original wild type phenotype which has been done by both true back mutation and suppressor mutation. Intergenic suppression. A suppressor mutation that occurs in a different gene is called an intergenic suppressor. Intergenic suppression is useful for identifying and studying interactions between molecules such as proteins. Proteins of biochemical signal transduction and gene expression pathways have been identified using this approach. Examples of such pathways include receptor ligand interactions as well as the interaction of components involved in DNA replication, transcription and translation. Here in the figure, you can see an example for intergenic suppressor where intergenic non-coding RNA acted as a new player in the p53 mediated repression of genes involved in apoptosis. In this figure, you can see the activation of p53 in response to stress triggers expression of 3 distinct classes of targets that silence gene expression in different ways, resulting in different cellular outcomes. The p21 protein inhibits cyclin dependent kinases, thereby activating the RB family of proteins and inhibiting the E2F transcription factors. This ultimately halts progression of the cell cycle. The microRNA or MIR34A degrades or inhibits translation of the mRNAs of genes important for both cell cycle progression and survival. A new class of p53 targets exemplified by the long intergenic non-coding RNA, lincRNA p21 is activated by p53 and then inhibits gene expression. Although the exact mechanism of gene repression is unclear, lincRNA p21 interacts with heterogeneous ribonucleoprotein K to downregulate a large group of genes that are repressed by p53, including several that are important for cell survival. The next mutation we are discussing is spontaneous and induced mutations. Spontaneous mutations are naturally occurring mutations and arise in all cells without a known cause. Spontaneous mutations arise from a variety of sources including errors in DNA replication, spontaneous lesions, and transposable genetic elements. Spontaneous mutations occur infrequently, although the observed frequencies may vary from gene to gene and from organism to organism. Measurements of spontaneous forward mutation frequencies for various genes of phage and bacteria range from about 10 raised to minus 8 to 10 raised to minus 10 detectable mutations per nucleotide pair per generation. For eukaryotes, estimates of forward mutation rates range from about 10 raised to minus 7 to 10 raised to minus 9 detectable mutations per nucleotide pair per generation. The smallest mutable fragment of the genetic material which serves as the basic unit of gene mutation is called muton. It forms a mutational site within a gene. The arrangement of mutons within a gene is strictly linear, just as the order of genes in a chromosome. A mutant is an organism or a new genetic character arising or resulting from an instance of mutation, which is a base pair sequence change within the DNA of a gene or chromosome of an organism. Mutation produced artificially by the application of non-biological agents is called induced mutations. The artificial induction of mutation is termed as mutagenesis, and the mutation inducing agents are called mutagens. Mutagenesis is a process by which the genetic information of an organism is changed in a stable manner, resulting in a mutation. Induced mutations are produced when an organism is exposed to a mutagenic agent or mutagen. Such mutations typically occur at much higher frequencies than spontaneous mutations do. The first induced mutations were created by treating Drosophila with X-rays. Using this approach, Mueller induced lethal mutations. X-rays, gamma rays and fast neutron were used to induce point mutations or deletions. Types of mutagenic agents. Mutagens are of two basic kinds, namely physical mutagens and chemical mutagens. Physical mutagens include different kinds of radiation like UV rays, X-rays, gamma rays and cosmic rays, and also the products of the decay of radioactive isotopes such as carbon 14, H3, sulfur 35, etc. Physical radiations are two kinds, non-ionizing and ionizing. The former includes short wave and high energy radiations such as UV rays, X-rays, gamma rays and cosmic rays. The latter includes alpha particles, beta particles and high energy protons and neutrons. The important chemical mutagens include base analogues, alkylating agents, acridine dyes, deaminating agents, hydroxylating agents, etc. Of these, base analogues act only on replicating DNA and the others act on both replicating and non-replicating DNAs. The absence of inadequacy of the DNA repair mechanism may lead to serious consequences. In human beings, it leads to an autosomal recessive skin disease called xeroderma pigmentosum. Alkylating agents such as ethyl methane sulfonate, ethyl ethane sulfonate and mustard gas can mutate both replicating and non-replicating DNA. By contrast, a base analog such as 5-bromouracil and 2-aminopurine only mutates DNA when the analog is incorporated into replicating DNA. Each class of chemical mutagens has specific effects that can lead to transitions, transversions or deletions. Let us now summarize what we have learned in this session. Mutation is a sudden change in a gene or unit of hereditary material that can alter an inheritable characteristic. Most mutations are not beneficial, since any change in the delicate balance of an organism having a high level of adaptation to its environment tends to be disruptive. As the environment changes, however, mutations can prove advantageous and thus contribute to evolutionary change in the species. Mutations are variously classified based on different aspects such as their origin, the type of cells in which they occur, the nature of the effect, their direction, intensity of the expression, and so on. Some of the major kinds of mutations are the following: somatic and germinal mutations, gametic and zygotic mutations, chromosomal and gene mutations, micro and macro mutations, biochemical mutations, lethal mutations, dominant and recessive mutations, silent and neutral mutations, back mutation and suppression, spontaneous and induced mutations. Here are a few questions for you to work out. 1. Discuss on chemical mutagens that disrupt DNA. 2. Discuss on mutation rates. 3. Elaborate molecular basis of mutation. 4. Discuss on H. J. Muller's classical work. 5. Discuss on model organisms in mutation studies. Here are a few books for your reference. 1. Genetics from Genes to Genomes, written by Hartwell L. H., Hood L. Goldberg, M. L. Reynolds, A. E. Silver, L. M. and Veris R. C., published in the year 2008 by New York McGraw-Hill. Human Genetics, Concepts and Applications, written by Lewis Rickey, published in the year 2001, 4th edition, Boston McGraw-Hill. An Introduction to Genetic Analysis, written by Anthony J. F. Griffiths, Jeffrey H. Miller, David T. Suzuki, Richard C. Lewontin and William M. Gilbert in the year 2000, 7th edition, published by W. H. Freeman, New York. Principles of Genetics, written by Gardner E. J. and Snustard D., published in the year 1984, 5th edition, John Wiley and Sons, New York. The Language of the Genes, written by Jones in 1993, published by HarperCollins. Here are a few websites for your reference: www.uvm.edu/till/cgep/education/mutations.html, www.wikipedia.org/wiki/mutation. Thank you for watching this program. Let's meet in the next session with a new topic. Till then, bye.